Jane and Charlie, if you have any comments or questions during the talk, please jump in. Yes, because we're such a small group that you can just interrupt me and ask right away. Right. Yep. So we'll just make it pretty easy. So here I go. I'm gonna start sharing the screen of my presentation, and then hopefully I'll do it right. Okay, that's screen. So you guys can see it, yes? Yes. Yep. All right, absolutely. So that's our introduction to super resolution microscopy. So my name is Stan Vita, and I've been in the Microscope and Imaging Center for a couple of years. And so today's presentation is really, I would say, introduction to super resolution fluorescence microscopy, because this is fluorescence is what all of these super resolution techniques are using. So but before we get to that, I have to tell you a little bit, you may know some of that already, but a little bit about the Microscope and Imaging Center, what we do, and or what we could possibly do for you. So we do sample preparation imaging, image analysis as a service or as a training or instruction. We have a number of light and electron microscopes for people on campus or even outside campus, but we also do consultation, troubleshooting, collaboration. So when people get trained, they're not on their own. We still are ready to help them. And we also offer some formal courses. So the Biology 608, the light microscopy course, has been around for about now 16 or 17 years. And this year, we're happy to offer, after a long break, an electron microscopy course, Biomedical Engineering 689, electron microscopy of biological and soft materials. They're both spring semester. So if you know anybody who would be interested in these, tell them so that they can sign in. All right. So, there are some super resolution microscopes in imaging facilities at A&M. And of course, there is a number of other super resolution microscopes in individual labs, but I only know of a couple, so that's why I'm mostly just mentioning the facilities. So our center has the Leica confocal system with STED super resolution. I'll be also mentioning the SRRF super resolution, which you can do on any microscope, and we also have deconvolution software for wide field as well as confocal images. Now, the image analysis lab in the vet school has a structured illumination system from Zeiss, and they also have the Zeiss Aeriscan system. There is also another facility, the integrated microscope and imaging lab in the Health Science Center, and I am not totally sure what all they have as far as going into super resolution, but they do have some fairly new instruments. So you can look them up and talk to them, with your needs. A brief outline, what all we'll cover. I'll recap some of the limits of optical resolution and why we may want super resolution. How does fluorescence work? And then what are some special properties of fluorescent probes that we would use for super resolution? And then there'll be a long list. There are so many ways to either to break the resolution barrier or at least to push it a little further. And I was going to actually, at the end of this presentation today, I was going to show you some live demonstration of the STED super resolution and then also of the SRRF super resolution from our microscopes, but we'll have to schedule the live demo maybe a week from now. We'll send everybody an announcement because we currently have some service done on that on the STED microscope. So I think it'll be, and even though, yes, I could show you some pre-recorded images, I think it's just going to be best when you're going to see it live as it's happening. So that we'll just schedule that for either next week or something like that. So this is a really nice link for a really neat review, fairly recent, this from Nature, regarding super resolution. And then just some disclaimers. I borrowed some of these slides or part of it from other people. And down here, there is your explanation of lots of things regarding fluorescence microscopy. So if you wanted to read up further on some of these, you're encouraged to do so. So the main issue with resolution using standard optics is that the resolution is limited, and that's because the image of a small point object is not a small point, but an airy pattern. So as you image objects, or say small point objects of decreasing size, initially the size of the corresponding image will be decreasing, but when you hit the diffraction resolution limit, even though your objects are smaller and smaller, their image of that, the image of that point remains essentially the same size, and it's an airy disk or airy pattern with central bright spot and then some concentric circles around it. And here is, if you see my cursor, can you see my cursor there?
Yes, we can.
Great,
yes.
Great,
yes.
Thank you. So this is your intensity profile if we drew a line through that airy disk. So it's a point. So if you have two small objects, so the airy disk is named after Sir Airy, an astronomer, British astronomer. So astronomers like to look at small pointy objects like stars, and that's what they encountered this problem first. So if you have two small objects far enough, you can distinguish between those two airy patterns, but as they get closer, at some point they get so close that you cannot tell whether it's one or two, and that's this is your resolution limit. And here is your equation for resolution, a standard diffraction limited resolution in XY. So roughly for your good oil immersion objective, we are looking somewhere around 200 nanometers, maybe, and it depends on the wavelength, something like 250 nanometers. And this is all dependent on wavelength and the numerical aperture, which is the most important number on the objective here. We don't really care much about the magnification on the objective. So that's your diffraction limited resolution. Of course, that's not good enough to resolve many interesting biological or other objects, because we have small organelles, we have microtubules and macromolecules or individual molecules that are all much smaller than the standard microscope can resolve. So what does it take to get the best resolution? So in the old days, in the 20th century, we really needed good lenses. because we were separating the neighboring features by focusing of light. And there the resolution is limited by diffraction. There is no way to tell certain features or molecules apart because everything overlaps on the detector. Yeah, you cannot make infinitely small array disks on the detector. In the 21st century, we went from focusing of light, which is diffraction limited, taking advantage of some molecular state transitions. And that could be transitions between the on and off state of fluorescent molecules. So what was, in the old days, what was a physics problem, the diffraction barrier, we now essentially have that as a chemistry problem. How do we make these molecules to transition between different states? And how can we take advantage of those states? So, but of course, when we do that and we increase the resolution, we have some new limits to what can be resolved. So one of them is how stable are those fluorophores? How many photons we can get out of a single fluorescent molecule before it's destroyed or before it's bleached. And that's typically a fairly important issue, especially with fluorescent proteins, because you get much fewer photons from, say, a green fluorescent protein than you would get from a synthetic green fluorescing dye. And that will affect your resolution. The other issue, of course, now we're looking at resolutions on the scale of nanometers. How stable is your microscope and whether the sample or the stage on your microscope is drifting. So there are all sorts of ways to correct that, but that's something that any super resolution technique has to deal with. And of course, one of the big limitations is how you also label your sample. I mean, this is something that's been known in electron microscopy for a long time, but if you take your standard indirect immunofluorescent labeling, you have your tissue with your target epitope, you attach your primary antibody, then you apply your secondary antibody, which has a fluorescent dye on it. And so your fluorescent dye, it could be localized anywhere within about maybe 50 nanometers away from the original target. You just don't know exactly which direction. So you're adding uncertainty this way. So there are ways to, typically people would then switch from indirect fluorescence to directly labeled primary antibodies, using also some of those newer small antibody molecules that allow better localization. But this is definitely something that you have to be aware once you start looking at resolutions in the order of tens of nanometers or nanometers. So a quick recap, maybe I should ask you some control questions. If you still remember how fluorophore work, but we'll leave it for the end of the presentation. You'll get a little test, okay? So how does a fluorophore work? So the fluorescent molecule of fluorophore absorbs a photon. Yeah, we excite it, we shoot a photon, gets absorbed, the molecule is in the higher energy state, it quickly internally converts this lowest energy singlet state, so it loses some energy, and then eventually it either emits a photon, and that's what we would detect as a signal, or it can decay in some other ways that does not involve a photon, which means we essentially lost that signal. It could also cross into the triplet state or the forbidden state, which is even lower energy than the singlet state. And then when it emits the photon, it's a totally different wavelength that we didn't even expect. So typically we cannot count on this signal either for detection. So and we can then repeat this cycle, and once the molecule is down in the ground state, we can excite it again, but it can only happen, a limited number of times before we destroy the molecule. And especially if you have oxygen present, those molecules do photo bleach relatively fast. So let's talk more about fluorochromes. So many of these super resolution techniques will require either special dyes that are highly photostable, or can be photoactivated or photoswitched, or we require particular conditions in the sample, in the mounting medium, that would encourage or induce some fluorophore blinking or transitions between dark and fluorescent states. Because many, or I guess maybe most fluorescent molecules can exist in two different states. One is two different forms. One is the fluorescent form, or we could call it the on state, where they absorb the photon of our excitation, and then they'll emit a signal. Here we excite with blue, we get a green signal. But the same molecule can transition into the dark state, where now it has a different absorption and emission. So when we try to excite with blue, we don't get any signal. And these, it can switch back and forth between those two states. It just depends how long the molecule can stay in the dark state or in the fluorescent state. So typically, high intensity illumination and presence of oxygen favors the dark state. And that's why some of these techniques that use these transitions have to include some reducing agents in the mounting medium to achieve just the right balance between those two. So some other molecules can be either photoswitched or photoactivated. So one example, for instance, is your photoactivated green fluorescent protein, which normally we would excite it with blue light, something like 488 nanometer laser. But before activation, it actually doesn't absorb in that range, and you're not going to get any reasonable signal. So what we do, we illuminate with activation wavelength, something like 405 nanometer laser, and that will convert that molecule from the off or non-emissive state, switches that or activates that, and now it's in the emissive state, so that when you excite it with the right wavelength, it'll give you a signal. And so this could be either permanent, so it's your photoactivated fluorescent protein will be in the on state until you destroy it or photo bleach it. On some other molecules which are photoswitchable, they can go back and forth between the emissive state, and then if we use a particular wavelength of light, we can switch them off and force them back into the off state. And some of these super resolution techniques are based on some of these transitions. So another phenomenon, see, we're covering all these boring theory parts here, so that then we can just zip through the rest of it. Another interesting optical or fluorescence phenomenon is stimulated emission. So let's see, we have a fluorescent molecule that got excited. So here we excite with something like 640 nanometer wavelength. We excite that molecule, and normally that molecule, when it emits a photon and goes down to the ground state, would give you a signal somewhere around 680 nanometers or something like that. But if we catch that molecule in the excited state, and illuminate that, hit that with a longer wavelength photon, 775 nanometer in this case, we will force emission of that at 775. So essentially, we're not going to get our expected fluorescent signal at 680. So we force it down to the ground state without emission of the expected photon. And that's part of the STED super resolution principle. So well, you'll hear about it a little bit more. So, but there are many ways to deal or get over the diffraction limit. So first, and I just listed them here. It's sort of categorized, but you'll probably find some more techniques that would fit in here, but it could go forever. So I limited it to these 8 groups. So first is just deconvolution or 3D image restoration, which gives you a slight improvement. So you couldn't really call it super resolution, but let's say from 250 nanometers, you get to something like 180, 170 maybe. And you also get improved contrast. Zeiss has their Eriscan system, which is an interesting application of in a confocal microscope. And they can get about 1.7x improvement in both lateral and axial resolution. So you're now down to maybe 140, 150 nanometers. And then there is structured illumination, which gives you about 2-fold improvement. So these, some people really don't consider these super resolution, but just a ways to push the diffraction limit a little further. And then we have other techniques, the pattern illumination with some nonlinear saturation phenomena, and that's the STED. One example is the STED system, and here we get down to about 30 or 40 nanometer in XY. There is also improvement in Z, which you could get actually about the same. The STED can go actually even to much higher resolution for some selected cases. Single molecule localization microscopy, that's a big group. And we'll talk about it in more detail. The examples include the palm storm, DNA paint, ground state depletion, and similar techniques that are, everybody likes their acronym. So, well, there's so many different names for this. Now, a relatively new technique, MinFlux, which is so far the highest resolution techniques, which is sort of a combination of patterned illumination and the single molecule localization. And this technique can get down to about 1 nanometer resolution. So you're now looking at molecular resolution. There are some other techniques. So these typically require specialized microscopes, hardware, and which means complications and money involved in there. There are some other techniques that actually you could do in your own lab on almost any microscope. And these techniques utilize the blinking or bleaching of these individual fluorophores. So that involves some acquiring let's say 50 or 100 images, and then looking at these fluctuations in the signal. The software can calculate the underlying super resolution structure. So it's either SOFI, SURF, PIMP microscopy, DSOS, and so on. So lots of names here. And yet another way to do super resolution is expansion microscopy. So if 2 molecules are too close to be resolved by a regular optics, maybe we can allow the sample to expand so that they get far enough, and then we can image them and resolve them in a regular microscope. So that's that technique. So here's a question to my audience. Which technique do you think would be best for really thick samples? Anybody?
Well, thick samples.
Yeah, really thick. Let's say you have a sample. Let's say you're trying to image something at high resolution, and it's say 100 microns deep in the in your sample.
I would think that area scan might be pretty good.
Yeah, because it's a confocal principle, so you block the out of focus light, so you get a much better contrast. Okay. That's true, and it's on a similar I guess level. It's also your STED microscope, because that's also based on the confocal system. Okay. So, okay, thank you. Well,
yeah, we are paying attention here.
Well, I'm just checking on you. So let's just quickly zip through the, these techniques. So deconvolution, it's also called a 3D image restoration. So the fact is, when you take a regular microscope or even a confocal microscope, your images are degraded by the way the microscope optics is representing a point object. Yeah, so in your images, you have some out of focus blur, you have aberrations, you have noise. So essentially your image is a convolution of the object itself with the way the microscope displays it, which is called the instrument point spread function. So that point spread function tells you how a point object is represented in the image stack or data cube. And so if we know the point spread function, we could try to reverse the process or estimate from our data how the original object looked like. So essentially, we're reassigning some of that out of focus light back into the position where it came from. And so this is an example. This is a single fungal cell multinucleate, which has a histone 2B tagged with GFP. And this was a stack of images, and in the raw image in the XY, you can see it's blurry, and you can see even worse in your XZ or YZ representation that there's lots of out of focus light. So after about 30 or maybe more rounds of deconvolution, you can get a much cleaner image with much less blur, and now you're able to resolve some of these smallish details in the nuclei. So far so good, but so deconvolution you can use for regular wide field fluorescence. You can use it also for confocal images. You get some increase in resolution. Mostly you get much better contrast, but it's not a method to take some awful images and feed them into software and get magically wonderful data. But it's good for imaging live cells because you can take some, faster, lower light images that are noisy, and you can get rid of some of that noise through the deconvolution, and typically your sample is not as photo damaged. It does not replace the confocal microscope, because if you have thick samples, let's say couple cell layers or maybe more than one cell layers, the blur is just too much and you cannot get rid of it. And it's also only good for like small distinct objects. So if you have some diffuse signals, deconvolution doesn't handle that very well. In the microscopy center, we have the auto Doppler deconvolution software for wide field images. So you can bring your data from your own microscope and process them here, and we don't charge anything for that. And on the Leica confocal, we have the Leica lightning processing that's integrated in the software. And that actually works quite nicely. So you could buy your own software. Typically, these are on the range of, $10,000 to $15,000. But they work quite well, and they're fairly fast in recent years. So that was deconvolution. The second was the Zeiss area scan. So on a regular confocal microscope, the detector, you have a single small pinhole, and so light that can pass through the pinhole represents the signal that's in the focal plane, and we rejected the out of focus light. We can get slightly better resolution even on the regular confocal microscope than in a wide field microscope. And if we close the pinhole down, the resolution would be increased. So you could close the pinhole down to say 0.2 of the size of the area disk. And that would improve your resolution, but you detect so few photons that the signal will be really noisy, and it may become totally unusable. Or you would have to illuminate for such a long time or with such a strong laser that you're going to kill your sample. So I solved this dilemma by using this array or this arrangement of highly sensitive detectors that are in this circular pattern. So each of these little spots represents the small pinhole, but they're detecting the signal in all of these. As you scan across the sample, they will record in each pass slightly different part of represent part of the image. And then we can reassign the phase and position information. So essentially you get high resolution as with a closed pinhole, but you also get lots of photons and really good contrast with that. So I think it's a really clever trick. Lately, a Breyer instrument has a somewhat similar system where they use a matrix of avalanche photodiodes to achieve something similar. All right, the other, so that's, I guess we could call it super resolution. You get to about 140, 150 nanometer resolution in XY. A similar level of resolution, it can be also achieved in some of those other confocal instruments. There is a spinning disk confocal super resolution microscope. This particular one is from Olympus, but there are other vendors. And it's a modified confocal system with some additional optics. And the advantage is that it's still a spinning disk, so it can be fairly fast. And you get instant, about twofold improvement in XY resolution, down to about 120 nanometers. So that's also a possibility. So that's, these were the direct, optical principles with those confocal systems. Another method is structured illumination, and that's based on the fact that if you have a sample that has some pattern that's too fine to be resolved in with regular optics, with diffraction limited optics, if you superimpose another pattern, in this case we project an illumination pattern. When those two patterns are overlay, the interference of those two patterns will give you a moire pattern, which we can detect because it has a much wider spacing. So we can detect that pattern, and because we know what our illumination pattern was, we can calculate what was the tissue structure. So The downside of this is that you have to take multiple images because you have to rotate your illumination pattern several times and also shift it so that you can get the increased resolution in all directions and also in the Z. So it's not terribly good for moving samples or live fast moving samples, and it involves post-computational, post-processing. So depending how you set your parameters, you can get either nice looking, less resolved image, or you can also get some maybe higher resolution image with some processing artifacts. So it's somewhat tricky to know exactly what you're getting. But the advantage is that you can use any fluorescent dye, just your regular dye. You don't have to do anything much special about the sample besides making sure that it is well prepared. But it's a wide field technique, so once the samples get thicker, it's not working that well because then you have a higher background, image blur. So, but you can buy these systems commercially. OMX, NSIM from Nikon, Zeiss has one, and this is the system that the image analysis lab in the vet school has. So you can get down to about 120 nanometers in XY, around 300 nanometers in Z. So, about doubling the resolution, so which sometimes is quite useful. Okay, so here is an example. Everybody loves the image microtubules because it's really easy to demonstrate how how well it works or not. So this is your regular wide field image, and this is your structured illumination image where you were able to resolve several closely spaced microtubules. So now we're getting to the STED super resolution. So it is an application or development for confocal microscopes. So given that it's a confocal system, we're already rejecting the out of focus light. So that gives you better contrast. But the limitation of a confocal system is that when we focus light into the excitation line in a single spot, but that spot is diffraction limited. So it cannot be smaller than about 400 to 500 nanometers in diameter. Yeah. So if we could get that excited spot smaller, we would have essentially a finer probe to scan across the sample and get much higher resolution. So the way it's done is that we excite the spot, and then we superimpose that longer wavelength laser that has a donut shape. So that it has zero intensity in the center, but it has some intensity around here, and that allows us to force those peripheral excited molecules from that spot down to ground state without photon emission. So effectively, we reduce the excited spot size here, and we can achieve much higher resolution. So that's essentially how it works. But of course, in order to be able to hit those excited molecules in that excited spot, we have to make sure that it's actually there's some good probability. So we have to pump a lot of that depletion laser light in it. We're looking at levels of like gigawatts per, in energy terms, gigawatts per centimeter square. So you can see already that that will probably cause a really fast photo bleaching of the sample. I remember in one of those first super resolution demos about 10 years ago, we were able to, I brought some slides with me, went to Houston, and we were able to almost take one image before it bleached. So it definitely, since then, a lot has improved in detectors and in the technology as well. But so the nice thing about this, it's a truly optical technique. So you don't really have to do any post-processing or further deconvolution or anything. And the other thing is that since you can change the depletion laser power, you can actually change or tune your resolution. The more power you use, the more you squeeze an excited spot and improve your resolution. Of course, the dimmer your signal will also be, because now you're collecting signal from a volume that's maybe like 100 times smaller than originally. So you'll get many fewer photons. But it's really nice. And if you come back next week, hopefully everything will work great, and I'll be able to show you that really on these modern systems, you just click a couple buttons and you go from regular imaging to super resolution. So the advantages is a confocal method. So it's you can use it for thick specimens. You can get resolution down to about 30 or 40 nanometers in XY. On our microscope, we can do the improvement in XY, but we cannot improve the Z resolution. But another about $100,000 would get us that option. Now, with some fluorescent molecules, which do not photobleach, like nitrogen vacancies in diamonds, they were able to get down to about 6 nanometer XY resolution. So these microscopes are commercially available. Leica has one, and the Barrier Instrument, that's another company. This is actually a company that was founded by Stefan Hell. That's the person who invented the STED and who got the Nobel Prize for it a couple of years ago. And they have, I think that's the cutting edge really instrument with lots and lots of other interesting features. So the downsides, high power, so you have photo bleaching. Only certain fluorescent dyes will work. So on our system, you need to use dyes that are excited around 650 nanometers. You can also use some that are excited by 590. So we could do 2 colors. We cannot do on our microscope, at least, we cannot do any fluorescent proteins except maybe like M Neptune, which is excited around 590. And it's a scanning method, so you image point by point. So it gets pretty slow if you need to image large areas. And those instruments are expensive and somewhat complicated because you have to keep everything aligned as well. So that was the STED, the scanning technique. So now we're coming up to the single molecule localization microscopy techniques. It's also sometimes called stochastic readout methods. So the problem, if you look down here, the problem with our diffraction limited resolution is that if those molecules or objects are too close to each other and they all are emitting photons, we cannot resolve it. Yeah? But if only some of them were emitting photons, they're far enough so that we can localize the center of that spot. And then if we could image another subset that would be also far enough, we could localize those. And if we repeat it enough times, we can build a localization map of that object. So that's so this is where we need those transitions between the dark state and the fluorescent state, and the ability to turn all of these molecules off and then activate just a couple of them. So the resolution in these techniques is typically around 20 nanometers in XY and maybe 50 in Z. there are some slightly better resolution variants, but that's kind of the typical level. So here is the same principle again. We turn all of these molecules off, or maybe they are off, and then we randomly activate some, localize them, we bleach them or turn them off, randomly activate another subset. We do this, a couple thousand times, and eventually we have this beautiful microtubule localization, map here. So two major variants or two major acronyms that you will see is PALM. This is using photoactivable molecules. So you turn them, you activate them, or some of them, and then you image them and until you bleach them. They're destroyed, and then you activate some other subset. So you image each molecule, each fluorophore just once. In the STERM system, it uses some special photoswitchable dyes or proteins, and those can be switched on and off repeatedly. And so but the overall principle is similar. So the good thing about these instruments, they're relatively simple because it's essentially a wide field microscope with some fancy lasers and cameras. This method offers a higher resolution than most other methods, and you can detect a large area, because it's all read out on a camera. But you need these special dyes, or you need to tweak your sample buffer conditions to get just the right rate of transitions between the on and off states. If you have too many fluorescent molecules or too few fluorophores, then it could be also a problem. And because you have to do many cycles of the activation and detection, if your sample has, if the object is moving, your sample is moving fast, it's not going to work very well. And it's a wide field method, so it's not terribly good for thick samples. But of course, you could nowadays use maybe a light sheet microscope and do that. And the localization precision is limited by the number of photons detected. Yeah, so if you detect only a few photons, you have a big uncertainty exactly where that molecule is. While if you can detect lots and lots of photons, you have much better localization precision. So this is a hard limit because molecule fluorescent dyes or fluorescent proteins have only a limited number of photons that they can emit before they are destroyed. So here we're limited by the number of photons emitted. In the STED system, I'm just gonna jump back, we're essentially limited by the number of photons that we can excite. Yeah, because if I have, if I can excite some fluorophore and then deplete most of it, then it doesn't matter if I only detect just a few photons as long as the background is very low. But the problem is that this whole scheme requires you to initially excite a lot of molecules. So that's your single molecule localization microscopy. Here is an example from an old paper of some microtubules imaged in a conventional wide field mode versus in the STORM system. And this is your resolution in this particular case measured as 80 nanometers, but it can be even better. And you can also do multiple colors. So that's PALM and STORM. Now there is a really interesting another technique in this group, and that's called DNA paint. So in the PALM or STORM, we use switching the fluorophores on and off, these transitions. Here in this case, if we have a freely floating fluorescently labeled molecule, it's moving too fast to be localized. But if we can make it stay for a certain number of milliseconds, then it can be detected and localized. And that's, you can take it as your on state versus the off freely floating state. So the way it's set up is that you have your target molecule in the tissue, and then you use your antibody to detect that, and your antibody carries a small oligonucleotide. And then you flood the sample with a complementary oligo that carries a fluorescent marker. So when the oligo is freely floating around, you cannot really detect it, but if it attaches to the complementary oligo on the antibody, it stays put for a short period of time, and you will detect it and localize it. And you can tune this on the length of the on time by the length of the oligo, design of the oligo. So you can, this is, I think it's just really cool. And the big advantage is that you can, you have the ability to multiplex, because you could initially apply 10 different antibodies against different targets. Each of your antibody will carry a different oligo. So you flood the sample with a probe number 1 that recognizes this oligo. And then once you detected enough molecules, you flood the same sample with your second probe that has a different, that will attach to a different oligo in your antibody number 2, and so on. So you can repeat it for 10 or more colors. People have built automated fluidic system, microscopes using Lego, and you can do this imaging on an automated basis. So you can do that on any microscope as long as you have a reasonable contrast. So wide field if it's thin, or turf if you need just a surface, or on a confocal, or using a light sheet. But the problem is that the probes that are just floating around, even though you cannot localize them, give you higher background. So you cannot use too much of a probe. And that means that your frequency of these molecules actually finding the complementary oligo and stay put for a short period of time is kind of low. So it takes a long time. There is an improved version that uses the fret paint that alleviates some of these problems. But you can read up on that if from this paper. But that's I think that's a really neat technique. So that was the fret paint. So that was now that takes us to the up-to-date, the highest resolution technique, which is called MinFlux. And this was developed in the same, by the same group, the Stefan Hell's group in Germany that invented the STED system. And they currently, the Aberrier Instruments is offering a MinFlux system. So what it means, or what it does, is trying to maximize your resolution or localization without emitting too many photons. Yeah? So they combine the scanning technique with the stochastic method. So they activate a spare set of fluorescent molecules. So they're just, every now and then there is a molecule. And then they use a donut-shaped excitation beam that scans around the emitter. And so, as they say in that paper, if we or a demon manage to target the zero of the donut excitation beam exactly, the donut targeting device would map the molecule in perfection without eliciting a single emission. So, you know, but since we are not a demon, we do have to get at least some photons. But the way it works is that we have an approximate localization of the molecule, and then we scan around, essentially triangulation around it, take 3 measurements or 3 excitations, and based on these intensities, you can determine exactly where the molecule is localized. So this technique, it's actually surprisingly fast, at least according to their videos. You can use it for ultra high localization precision. Here is an example down to about 1 nanometer. So your single molecule localization, you know, your Storm or Palm was unable to resolve this spot, but MinFlux tells you that yes, you have 2, 4, 5 different emitters in this spot. Here is the schematics, simplified schematics of that microscope. It's extremely complicated, it would seem, and but they do have a description of it here in the products. But it seems like it's a really neat technique that you can use for localization. You can use it also for tracking of individual emitters, either over short distances or even over some long distances like micron travel. So and this principle seems to be like a really a breakthrough in resolution. So these are great, but not everybody has access to these instruments. And there is a number of other techniques which you can essentially do on any microscope that you have access to. You may not get as much resolution enhancement. You won't get down to 1 nanometer, but one such technique is SRRF, SuperResolution Radial Fluctuation Microscopy. So this is based on the principle that if you have multiple emitters, multiple fluorophores that are close to each other, and when you image them, they just look like a blob, like a diffraction limited spot. But if you take a series of images, some of these emitters maybe will, as they're blinking, they'll go into the dark state, and some will remain on, and that will slightly change the radial symmetry of that spot. And the analysis of these patterns will allow you to determine this underlying pattern of fluorophores. So you can get down to about 80 nanometers, and there is a free ImageJ plugin, so you can, free software, you can download that and test it yourself. So there is the plugin has detailed instructions how to use it. So that's one good option, and here is an example of that, where they were able to get, again, imaging microtubules, the favorite object, down to about 80 to 70, 90, or around 100 nanometers. And so, but typically you need to take about 100 images. So if your sample is moving fast, it's not going to work well. But the advantage is that if you take many images, you can decide after the fact really how many frames you'll be processing as one time frame. So you can take 300 frames and process it as like 3 100 frame images, super resolution images, or you can take it as 12 frames, with 25 frames each, which will have less resolution in space, but more resolution in time. So you can slice it, this way or that way. So that's SRRF. Some Andor cameras actually have this processing built in, so they give you the super resolution image right out of the camera pretty fast as you're imaging. Another somewhat similar, somewhat older technique is photo bleaching microscopy with nonlinear processing, and the authors called it PIMP microscopy. And instead of relying on the blinking of these individual fluorophores, what they do, you again take a series of images, but you use sufficient illumination intensity that you're actually going to bleach some of these fluorophores. So as you keep imaging, some of these fluorophores will get bleached, and that will slightly change the shape of these diffraction limited spots. Again, it calculates the sub diffraction pattern, and there is yeah, and gives you a higher resolution image. So I have the little asterisk here because last time I used this method was about 8 years ago. So I just earlier this week contacted, and so I had access to their server for free processing, and they're since then I wasn't sure what's happening, but apparently there is still access. So you know, you could contact the author here and ask for your own access to it. I can report hopefully next week how well these images look once I process a fresh data set. So, and here is an example of the PIMP microscopy. They have a wide field image, and there is a storm image, and your PIMP image here is somewhere in between those two techniques. So you get down to maybe about 80, 90 nanometer resolution, and you can take images from a wide field system or a confocal. So both of these, the SRRF and the PIMP, unfortunately need a big number of images. So if you need to do live imaging and things are moving, that's problematic. Luckily, you have another technique, which is called DSOS. And the principle is that you just take 2 images at a slightly different excitation power, and then you just process those 2 images and get about 2-fold resolution. But what you need to use is a confocal or multi-photon microscope. The software is free. There is a Windows executable, or if you have MATLAB, you can run the MATLAB program, and there is a link there. The authors claim that it's better at resolving complicated structures than the SRRF method. So you'll have to see how that is. So, and that wraps it up. We are already over time. Did I lose anybody? Oh, we have another user. Hello, Drew. Ooh.
Hello.
So welcome. Sorry, I didn't watch the list of participants, but and I don't know when you joined, but if you have any questions, jump in and interrupt me.
Sure, it's quite physics. I'm not from physics department, so I'm from Ping Hei's lab. You know, yesterday he contacted you. Yeah, I'm just want to know how it's going with this super resolution technicals, and which might be useful for us because we also want to track some single molecular dynamics. Yeah, just kind of, yeah, it's really useful for me.
Okay,
I know.
Well, good to hear. So
thank you.
Sure. Okay, so I think the last method on my list here is the expansion microscopy. So, as I said, if your fluorophores or objects in your sample are too close to be resolved, we could try to actually just force them, separate them. trick is to make sure that the relationship between those individual structures or molecules of interest remain the same after you allow the sample to expand. But the principle is that you can convert the samples to like a hydrogen matrix where the sample components, whether it's proteins or mRNAs or other molecules, are cross-linked to the hydrogen matrix. And then you allow the matrix to swell. And as it's swelling, it's taking those cross-linked components with it. So now they will end up physically separated and can be actually resolved on a standard microscope. It used to be that you could swell the with the existing hydrogels about 4 times in all directions. So that gives you a volume about 64 times larger than the original. So you get about 4 times improved resolution. So around 70 nanometers. Recently, apparently some of those new gel formulations allow you to swell the sample up to 10 times and it's still linear. So you could get down to what, 20, 25 nanometers, maybe something like that. There is lots of information on this expansion microscopy website, and there are some references also down here. So that's the expansion microscopy. And I think we're almost at the last slide. So that was a run through all of these different techniques. Some of it require fairly high-end instrumentation, some do not. So you know, there's many different ways to get past the diffraction barrier. And it depends also on the sample that you're trying to do. There is an example of the expansion microscopy and the whole outline. I think everybody, we will have, or maybe Ashlyn will email you the PDF version of this presentation so that you can look at it, you know, later after the fact and look at some of these references. So, and that's the last slide. So the question is, you know, now that we can do all this fantastic resolution, can we still get away with some bad samples? And it's unfortunately, you know, you have to get a much better structural preservation, and the whole sample preparation pipeline has to be much more critical, because now that you're looking at maybe tens of nanometers resolution versus hundreds, then the artifacts and sample degradation that you couldn't see before may become obvious. So that's my final word of caution for you. We will not do the practical demo of the STED system today because we have a Leica service engineer working on the system. But I'm very optimistic that next week, the same time, you'll be able to do it. So we'll send updates, but Ashlyn will be sending you guys a link to another Zoom meeting when that happens, and so. But you can pencil it in on your calendar, I think, at least for now. We'll let you know if there's any further delay on that. So now I'm ready. Well, first I'm ready for your questions, and then are you ready for your little quiz?
A quiz?
Of course.
Well, what apple tastes the best? What is it? A food quiz? Is it a lunch?
No. I mean, I'll need to ask you some questions to see if you were paying attention.
I see.
But maybe you'll have some questions for me. I mean, that's one way to deflect my quiz if you ask me a question.
I see. Okay. Ugh, so looking at what you presented, Doc, for me as an embryologist, you know, I just basically use the confocal over at the image analysis lab. But if I wanted to step that up a notch, then I would need to probably, I prepared everything tunnel. I would have to go to maybe a different format to say go to the next level of imaging. Okay.
Well, you could consider the expansion microscopy.
Yes.
Because that's, you know, that mostly involves, you know, wet bench work, and then you use the same microscope on the expanded sample.
Right.
And, you know, hopefully get a much higher resolution that way. I mean, that's one way to do it.
Okay.
The other, you know, the other would be, you know, for the STED system, on our machine, you would get the XY, if you use the right dye, which would be something, you know, something like, you know, 650, 640 nanometer excitation, or 590. That would give you XY super resolution, but not in Z. So that you would still be limited there in for a regular confocal resolution. Right. So there's how big are your samples?
Well, initially they're the size of the embryo, and so we look at the embryo structurally that way.
Yeah, but can you translate it for a plant biologist?
Oh, okay. We're looking at 150 to 200 microns.
Okay, so fairly big in terms of super resolution.
Yes.
So yeah, I would say either that, or you could, you know, start contemplating the like the DNA. If the DNA paint or the, you know, the fret paint method would be applicable, that's something that might be possible, taking advantage of the light sheet system. But that's, you know, we have never tried that.
Right.
So that's something that would be really interesting. But you know, it's not like I cannot just hand you a protocol and you follow it and everything is gonna be great. So,
that's how it normally works out, Doc.
Oh,
unfortunately.
Well, not this time.
Okay.
So.
All right, Doc, thanks. Did I pass?
Yes, you did.
Okay.
Did. I won't ask you any more questions. All right. And any other burning questions that you always wanted to ask, but, you know, were afraid to, regarding microscopy?
I have one more. I have one more, Doc.
Yes, please.
We work on zebrafish, and we're looking at brain neurons. And if we were to get a live sample to actually see neuron processing, say, for instance, nanodiamonds between the neurons, it'd be a moving type of, what kind of microscopy would be best for that?
So I think for moving things that go fairly fast, I would say either looking at light sheet, if that's you know, if your sample is transparent enough, because that can you know, I mean, Holly will be able to tell you more if she's still on.
I think she got off.
Okay, but anyway, Holly Gibbs from MIC is doing light sheet microscopy, so she might be able to tell you more specifics in that.
Okay.
And
we'll get back to
that would be able to combine your, you know, fast volumetric imaging with hopefully some reasonable localization of small emitters.
Right, right.
If you can do it right.
Okay, thanks.
But maybe other people have different ideas. So yeah, go ask some other people too.
Okay.
All right. Well, is anybody raising their hands? I guess I can stop sharing my screen so that you can see me again.
All right.
All right. No other questions? Well, if you do have questions or want to discuss your samples without anybody else listening, you can just let me know and we can set up individual Zoom meetings where we can see what if anything we can do for you in those terms, because yeah, we do optical microscopy, we do electron microscopy too, so all sorts of possibilities. All right, well, if there's nothing else, no other questions, I thank you for your participation and attention, and just let me know if you need anything.
All right,
And
thank you, Doc.
we'll see you next week, maybe.
Lord
Thanks,
willing.
Dan.
Thank you.
All right.
Thanks, Ashley.
Thank you.